Hello, my name is Rick Farouni, and I'm a postdoctoral researcher at McGill University in Montreal, Canada. The title of my talk is Model-Based Analysis of Sample Index Hopping Reveals its Widespread Artifacts in Multiplex Single-Cell RNA Sequencing. Allow me to unpack the meaning of some of these words. Multiplexing. Whereas before we would run each sample on a single lane of a short read sequencer, nowadays we pool and sequence cDNA fragments of several samples in a single run to utilize the massive capacity and cost efficiency of the new platforms, such as HiTSeq 4000 and NovaSeq 6000. This comes at a cost, and this is known in the form of an artifact known as sample index hopping. In sample index hopping, the sample barcode of a given sample is misassigned to another sample. So, for example, here, the sample barcode that we assign to reads from sample B1, one of them hops to sample A1, and this shows up as a phantom molecule associated with gene SFN. We make 4 contributions in this paper. The first contribution is the molecular proportions complexity profile that allows us to characterize the library complexity of a set of samples and allows us to determine which samples are characterized by low complexity due to bad library preparation or self-collaboration RNA content. We use the molecular proportions complexity profile as well as parameters in the model, as we show in the paper. The second contributions we make is a generative model that characterizes the phenomena of sample index hopping. And this picture of multinomials model assigns a probability to all events that we see in the data, whether these are observations that show a collision that indicates a hopping, or observations that don't show any collisions. We call these observations non-chimeras. The problem that the model needs to solve is to determine which reads are hopped and which molecules are phantom. For example, observations we call in the model as Chimeric Fugues. There are observations where all the reads from a given sample hop to other samples. For example, 3 reads from sample 1 hop to samples 2 and 3, and this gives rise to 2 molecules, phantom molecules in sample 2 and sample 3. We make 2 assumptions in the model. First assumption is that all sequencing reads with a given cell barcode, a CUG, can be indexed only by the same sample barcode. And the second assumption is that the probability of hopping does not depend on the source or target sample. This allows us to derive a closed form expression of the distribution of sum of non-chimeras, and which allows us to estimate the sample index hopping rate using a generalized linear model with a log link function. As we see here in the top plot, the trend for non-multiplex data is flat, whereas the trend of proportion of chimeras for multiplex data it increases as a function of reads per CUG, and by fitting the model to the data, we can determine the index hopping rate. We use the index hopping rate and the proportions, the MPC, to find the distribution of the sample of origin, and using the argmax and the distribution of the argmax of the true sample of origin, we can choose three strategies. One, we keep the data as it is. Second, we reassign reads to their inferred sample of origin. We don't discard any molecules, or we discard molecules based on a cutoff that we determine by the trade-off ratio that we describe in the paper. If we don't purge, as we see here from the figure, the false positive rate is up to 11,700,000. While if we reassign, we can reduce the false positive rate to almost 700,000, only with a slight increase in false negative. And we can produce a trade-off, an optimal trade-off that balances both the false positive and false negative using the trade-off ratio. The impact of index hopping on downstream analysis can be shown by this figure, where we see a whole cluster of cells that was reclassified as empty after purging. And on the right, we see the effect of purging on gene expression profiling in a validation data set with known ground truth. There are two main points I would like to make. One is that phantom molecules can lead to complications, and multiple complications in downstream analysis, from misclassification of droplets to emergence of phantom copies of cells. But most importantly, even when the sample index hopping rate is small, it can give rise to a large fraction of phantom molecules in low complexity samples, as this table on the right shows. We implemented the procedure in an R package called Phantom Purger, and we have provided reducible analysis code notebooks for all the analysis in the paper, and they're available at the paper's website. Thank you for